Okay, so I would like to welcome everyone to this edition of the RNA Collaborative Seminar Series. My name is Eric Wagner, and I, along with Dave Matthews, are the Associate Directors for the Center for RNA Biology here at University of Rochester. The Center for RNA Biology has been active here for the greater part of 20 years and is directed by someone I think many of you know, Dr. Lynn McQuat. The Center for RNA Biology here has actually been one of the founding members of the collaborative seminar series. Back in 2020, there were only 4 members at that point, but I'm proud to say that now we have 27 members of this seminar series, with the newest one actually joining this week at Ohio State University. So I want to welcome those folks. So before I introduce our first speaker, I want to remind you all that to ask questions, please use the Q&A feature on the bottom of your screen to type your question in. During the question and answer portion at the end of each talk, I will reread those questions, and then the speaker, of course, will answer them. And the purpose is that these talks are recorded and the written questions are not visible on those recordings. So without further ado, I would like to introduce our first speaker, Dr. John Lueck. John is an assistant professor here at the University of Rochester in the departments of Pharmacology and Physiology and Neurology. The title of his talk is Development of Therapeutic Anticodon Edited or ACE tRNA Technologies for Nonsense-Associated Diseases. All yours, John.
Awesome. Thank you. Can everyone see my slide? Everyone can hear me? Now that you've gone, Eric, I can't get the thumbs up, but I'm just going to
You're all good. Yes.
All right. I'm going to assume that everyone can see it. All right. So thanks for having me. I'm excited to share our recent work on the development of therapeutic anticodon edited tRNA technologies. We shorten that up to ACE tRNAs. So a lot of what I'll be talking about today is unpublished, but hopefully soon to be published work. So to dive right into it, what is the problem? Problems are nonsense mutations. 10 to 15% of all genetic diseases are caused by nonsense mutations or premature termination codons, also referred to as PTCs, that affect about 3 million individuals in the United States alone through 7,000 distinct genetic diseases. Nonsense mutations arise from a single nucleotide mutation that converts a canonical codon into one of the 3 stop codons, TAG, TGA, or TAA. Nonsense mutations in general have more serious ramifications than missense mutations because you have almost a complete loss of the gene expression. And in some instances, actually, the truncated protein can have a dominant negative effect. So we all are probably familiar with many of the diseases that result from nonsense mutations. I've listed here a couple of kind of the more prominent ones: Hurler syndrome, beta thalassemia, Usher syndrome, spinal muscular dystrophy, Duchenne, and then the family of retinopathies, and cystic fibrosis. And so I'll be talking a lot about cystic fibrosis. This is the disease that we're mostly focused on in the lab. So because of the exceedingly high prevalence of nonsense mutations, so it doesn't really matter what gene it's in, the mechanism is always the same. It's a unifying mechanism where you convert a codon to a stop codon, a sense codon into a stop codon. There's a concerted effort to identify PTC therapeutics. So for cystic fibrosis, the name of the game really is to increase the amount of CFTR protein expression. So in cystic fibrosis, or CF, there are about 6 classes of mutations, and there's actually already therapeutics that are available through Vertex Pharmaceuticals that will work for about 90% of individuals with CF. However, they all work on the protein. So you have a class of small molecules called correctors, and I've listed 3 of them here: lumacaftor, tezacaftor, and lexicaftor. And they work through rescuing protein folding, processing, and trafficking. There's also a class of small molecules called potentiators, and that's known as ivacaftor. And so this is for channels that actually get to the surface membrane of the airway epithelial cell, but they don't function properly. They have a low open probability. And so ivacaftor binds to the CFTR channel and increases its open probability to allow chloride to move through to the extracellular space and also bicarb. However, there are no drugs currently for class 1 mutations, and those are mutations that arise from nonsense mutations because there is no protein that is actually generated. So taking a look at nonsense mutations, we think of this as a double hit in that this is a very generic schematic of protein translation. In normal translation, obviously, the ribosome moves down the mRNA, and this green cloud is a full-length protein. With nonsense mutations, the ribosome stops at the stop codon. You have truncated protein. Obviously, that's not functioning. But also, you have degradation of the transcript through nonsense-mediated decay. So without the full-length protein as a therapeutic target, the therapeutic focus is shifted to CRISPR, targeting the genome, total gene replacement, and actually targeting nonsense-mediated decay processes to increase the amount of transcript present for spurious read-through of nonsense mutations to hopefully hit a therapeutic threshold of protein amount, and then also focusing on translation processes. The approach that we're taking is different. We're using suppressor tRNA approach, and we call this anticodon engineered tRNAs. So regularly, tRNAs are recognized by the amino acyl tRNA synthetase, and they're charged with its canonical amino acid. That charged tRNA is then recognized by the EF1 alpha protein, and we think of this as like a helmet for the acylated tRNA. It's important for keeping the tRNA from being deacylated, but it's also important for delivering this charged tRNA to the ribosome where it can participate in translation. When the ribosome hits a stop codon, there are no naturally occurring tRNAs for stop codons, and so translation stops, release factor comes in and dissociates the ribosome and stops translation from occurring. Our approach is to take a naturally occurring tRNA, editing the anticodon now to recognize one of the 3 stop codons, where it is still recognized by the amino acyl tRNA synthetase, charged with this canonical amino acid, recognized by EF1 alpha, and brought to the ribosome where it participates in translation when it comes across a stop codon. So the success of this approach really is how much of this acylated ACE tRNA is present in the cell, because you're in competition still with the release factor. And so this is a competition that's a little bit different than maybe many people think about. It is whatever gets to the ribosome first wins. So it's like a horse race. So if you have enough tRNA, ACE tRNA that's present, and it outcompetes in that it gets there first, the release factor will not come in and dissociate the ribosome. And I'd like to be clear here that people call this technology suppressor tRNA technology. We call it ACE tRNA technology. And the reason we do that is because suppressor tRNAs occur naturally. And this is a purposeful mutation to a tRNA. So we've engineered them to be such. I'd also like to point out that this is not our original idea. We're taking an idea that has been investigated for many years. In fact, originally it was investigated by Temple et al. In 1982 for using suppressor tRNAs as a therapeutic for beta thalassemia. So our approach is to take the existing knowledge and to hopefully get it past the goal line for it to be a therapeutic. And important here to point out is that with all of this machinery, the only thing that we need to deliver as a therapeutic is the tRNA. And so tRNAs have favorable characteristics. The entire tRNA gene is actually only about 155 base pairs in length. The tRNA itself acts as its own promoter. And so delivering the tRNA is actually feasible using virus and small DNAs, which I'll talk about later. You can also deliver tRNAs as RNA itself, and there's lots of groups that are working on that. And why we think that this is important is that diseases that occur in different tissue types will probably take a different delivery approach. So like for the lung, where it has access to the outside, you can actually use probably all 3 of these approaches. You can use RNA, DNA, and virus. However, if you wanted to go after nonsense mutations that occur in the muscle, cause disease in the muscle, likely going for a DNA and viral approach is more favorable. So the genetic code has a bunch of tRNAs. When we originally started this, I was a postdoc in Chris Ahern's lab at the University of Iowa. We knew nothing about tRNAs. And so we just thought, okay, we're going to go in and we're going to find some tRNAs, mutate the anticodons, get our tRNAs to move forward. We found, and as many of you know, there's many genes that encode tRNAs in the human genome. There's about 415. The amount of tRNA genes actually differs greatly between organisms. Remarkably, bush babies have about 45,000 tRNA genes in their genome, and we actually have no idea why that is the case. And then there's lots of groups that are working on this. And I always say is that the reason that I think that bush babies, which are these animals here to the right, have so many tRNA genes is that they're really important for the expression of cute genes and their adorable large eyes. So what we did is not picking one tRNA from the human genome and moving forward. We came up with a way to screen all of them using this high throughput cloning and screening approach. So we have a nanoluciferase, which many of you are familiar with, where the last about 10 amino acids need to be present for the protein to luminesce. So we developed a screen where we put a stop codon before the last 10 amino acids, put it into a plasmid where we also can high throughput clone in our tRNAs of interest. So the amount of stop codon suppression will be measured in light in a plate reader assay. And we went through all 450, all unique human tRNA sequences, and we screened them all for their ability to suppress a stop codon. And these are all shown here. On the x-axis is the log scale of luminescence, and we've broken them down into different families. And one thing that we were pretty surprised with is that not all tRNAs can actually be converted into an ACE tRNA. Glycine really kind of highlights that, where many of them have no function at all, and some of them you can see have pretty good suppressor tRNA function or PTC read-through function. Then you have families like leucine and serine, where you have many winners, and they have really high suppression efficiency, and you can actually suppress all three of the stop codons. So what has been done? Using ribosomal profiling, we determined that ACE tRNAs suppress natural termination codons at low efficiency. And this was done in collaboration with Bill Skatch at the Cystic Fibrosis Foundation Therapeutics Lab. And this has been shown again after our original study. Using mass spectrometry, we determined that ACE tRNAs encode the intended amino acid. So this results in homogeneous and uniform protein expression, and in this case, CFTR protein expression. So we can get back the wild type protein. ACE tRNAs are effective at suppressing PTCs and rescue near wild-type CFTR transcript abundance and channel function when the transcript is encoded from the endogenous gene. And so this is really important, as we discussed, that with a nonsense mutation, you get NMD. And so one of the concerns is that there was not enough transcript for these ACE tRNAs to work on. But we've shown that by delivering these ACE tRNAs, you actually are able to rescue transcript abundance. So what are we trying to figure out? Can one ACE tRNA sequence be used for many CF nonsense mutations? And this is for actually getting a therapeutic. There are dozens of nonsense mutations that result in CF. And if we were to use different sequences of ACE tRNAs for each one to try to get them to be a therapeutic, this would be a nightmare getting through FDA. So one of our goals is to find one sequence that works for the majority of these nonsense mutations and bring it forward. Can we make ACE tRNAs more efficient at PTC suppression through engineering? And can we effectively deliver ACE tRNAs to the target tissue? So for this talk, I'll be talking a lot about a cell model for cystic fibrosis. And these are a human bronchial epithelial cell line that have been immortalized and then genetically engineered using CRISPR to have common CF mutations. And these are a great tool. These are developed by Hillary Valley at the Therapeutics Lab, where you can use them to measure the rescue of CFTR transcript abundance, CFTR protein, and most importantly, the CFTR channel function. One thing I'd like to point out here, is on the right, is using an approach that's very similar to like an Using chamber experiment, which measures the amount of CFTR function, which is depicted here by this deflection up from baseline is the CFTR current, that with a read-through agent, so aminoglycoside G418, with a inhibitor for nonsense-mediated decay, SMUG1, and also in the presence of the potentiator that I described that Vertex has developed, VX770 or ivacaftor, the most amount of rescue that was able to be obtained in W1282X mutation is 2.7% of wild type. So what this means really is that rescuing CFTR protein function is quite difficult. So what we want to determine first is if we could deliver our ACE tRNAs to these cells, what is the maximum amount of rescue we can get? So we used a whole cell patch clamp approach, and we used R1162X mutation and W1282X mutation. And we transfected in plasmids that have our ACE tRNAs with also a neon green, so we could see which cells were delivered to. We'd pick the green ones and we would patch them. We would do a ramp protocol from a negative potential to a positive potential, and we'd plot the current elicited at plus 80 millivolts over time. And that's what I'm showing here from wild type HBE cells. The channel is activated with forskolin and IBMX, and then there is a quasi-selective inhibitor 172, and you can see the decrease in CFTR function. And so the difference between the activated and the blocked is the inhibitor sensitive current. So this will tell you how much CFTR activity you have. And in the W1282X cells and the R1162X cell, you see that you have no current. We then picked the green cells. In red are 1162X that had received the ACE tRNA that's suppressed with an arginine. And you can see compared to wild type that we get about a 75% rescue in current. And in blue, rather than putting in a tryptophan, we put in a leucine. And this is mostly because our tryptophan ACE tRNAs aren't very active. And David Bedwell at University of Alabama, Birmingham had shown previously in studies he had done with aminoglycosides that when leucine was incorporated at that position, it gave near wild type level. And so of channel activity. So we used our leucine suppressor tRNA, and we showed here that we could rescue the function near 100%. Okay, so we became a little bit more sophisticated, and we wanted to do more high throughput studies. And so we transferred from, rather than doing transfections, we used adenovirus. And when you transduce the 16 HBE cells on plastic, you can see there's a dose-dependent increase in transduction efficiency near wild type. But in order to do these Using experiments where you're measuring CFTR function, you actually have to plate these cells in transwells. They form tight junctions, and this takes about 7 to 8 days. When you transduce the adenovirus, the best transduction efficiency that we can get is about 25 to 30%. And so that's important for the studies that I'll show here. So this is a busy slide. We'll start at the top left here. In red, in R1162X 16 HBE cells, and we're looking at transcript abundance here with a scrambled ACE tRNA, the wild type cells, and then wild type cells that have received the arginine ACE tRNA. Compared the amount of transcript that's rescued with an ACE tRNA suppressing with an arginine, you can see that we get about 50%. Looking at function, you're looking at Using traces. This is wild type in black, that we rescue a moderate amount of the ACE of the CFTR function. And this is in the presence of lumacaftor, elexacaftor, and tezacaftor, and ivacaftor, just to see if we can augment the amount of current that we get rescued with the ACE tRNA. And that's represented over here on average. And so in the red bar going across, you can see that this is the transduction efficiency, and that we're rescuing almost to that point, like maybe 75% of the current in those cells that have received the virus. So what this means is we're able to get our ACE tRNAs into 25% of the cells, and of those cells, we rescue about 75% of activity. So this is a delivery problem. With W1282X mutation with leucine, again, we rescue about 50% of the transcript abundance, but the function is far better than that for R1162 mutation. And it's a significant and robust rescue of CFTR function. And again, we're rescuing protein in which the Vertex drugs act on. And you can see that we actually super rescue the W1282X in the presence of ETI, which is Trikafta, which is the approved FDA drug for Vertex, that Vertex has produced. Okay, so next we wanted to see is if we inhibited NMD on top of using an ACE tRNA with these correctors, can we increase transcript and channel function? And just to summarize all of this, the answer is yes, that if you also, on top of using an ACE tRNA, inhibit NMD using SMUG1 inhibitor, that you do get a modest increase in function here when in the presence of SMUG1, but it's much more effective NMD inhibitor in the presence, and for W1282 in the presence of ACE tRNAs. So, and this is a dose dependence of the ACE tRNAs. So what we're showing here, that in the presence of SMUG1 and with ETI, that you need actually very little, an MOI of 10, to rescue the same amount if you would need an MOI of 500 with just ACE tRNA alone. Okay, so what I've shown here so far is that we can put a in a W1282, we can use a leucine to effectively rescue wild type levels of CFTR function. And so we wanted to expand this idea, is that, okay, if leucine works for W1282, can we use maybe arginine for all of the most common mutations, or leucine for all the most common mutations? And so that's what we did. We actually took all of our 16 HPE cells that have been genetically engineered and tried each of our ACE tRNAs for each of those mutations. And so G542X represents about 2.5% of individuals with CF, R55 3X 1%, 0.4% for R1162X, and W1282X represents 1.2% of individuals with CF. And so here is looking at, just in a bar graph, the functional rescue of CFTR. And so we've broken it up into using color into the different mutations. So a G542X, which is the most common, rescued with an arginine ACE tRNA in the presence and absence of Trikafta, those Vertex drugs. We rescued with the glycine ACE tRNA and leucine ACE tRNA. So just looking at this panel, you can see that using a glycine ACE tRNA rescued moderately. And again, leucine was able to work very effectively at this position. Arginine worked very well. Glycine did not, and leucine did. So effectively what we're making here is a G542R and a G542L. Glycine didn't work because, again, that ACE tRNA is not very effective. So this pattern really is the same that you see for all these mutations. So arginine and leucine was able to rescue functional CFTR in 4 different mutations. And again, this is exciting. Leucine is being the best. We think that we can now bring leucine forward, and we're going to test this for many more CF-causing nonsense mutations. We did this in a different cell type, and so this is a model that the Cystic Fibrosis Foundation Therapeutics Lab is developing. It's patient-derived intestinal epithelial monolayers. We call them HPEMs. And using an assay that I've discussed before, looking at CFTR functional rescue, this is just the representative traces here, and this is the average traces on the right. So this patient had a G542X, R1162X compound heterozygote. And so we rescued with G418, which is kind of the gold standard for aminoglycosides. And you can see that it rescued a modest amount, but with increasing amount of ACE tRNA, you can rescue a significant amount of CFTR function. And with the Trikafta, in an MOI that is much lower, you can rescue a significant amount. So again, this is a combinatorial therapy. So to summarize this bit, we've shown that one ACE tRNA works for many or most CF-causing mutations, and that leucine is kind of our front runner here, and we're currently testing this with 30 other of the most prevalent CF mutations. And we're excited about this. We're also going to go forward and, through collaboration and at my lab, look to see if one ACE tRNA can rescue nonsense mutations that result in produce Duchenne titinopathies, dysferlinopathies in a pulmonary disease, primary ciliary dyskinesia. So now that we know that this works, we wanted to see if we can take an ACE tRNA sequence and optimize it for better function. So optimization for transcription, its post-transcriptional processing and folding, and maybe even its recognition and charging with the aminoacyl tRNA synthetase. And so lots of groups, including the company tRNA, are using artificial intelligence to do this. We're using a method that's different. We call it PI. And this is Porter intelligence. So this is Joe Porter in the lab. He's a postdoc who's really done a deep dive in doing nucleotide substitutions in ACE tRNAs. Also, the members of the lab refer to him as ChatJoePT. He's a very talented scientist. On the left, you see him holding a bottle of prototelomerase. Here, it goes on the market. It's very expensive. We wouldn't be able to use for the experiments that we do, actually purchase this from companies. It's prohibitively expensive here. He's generated in a weekend $1.2 million of prototelomerase, and he's obviously very excited about it. He's a connoisseur of many things, including wings. So what he's done is he's gone through the sequence of the tRNA gene, and he's looked at the upstream control elements, the trailer, which is important for transcription termination and processing. He's also gone through and made substitutions within the tRNA body and to see if he can improve folding and recognition by the amino acyl tRNA synthetase. And he's generated this like really quite beautiful screen method using the same kind of nanoluciferase with the stop codon, and he's able to screen over 2,000 tRNA variants in this way. And so what I'm showing you here is the rescue of CFTR protein. So this is CFTR that is transfected into cells, and it has a nanoluciferase on the C terminus. If anyone's ever done Western blots on CFTR, they know that they're quite difficult. And so we wanted to create an assay where we can do in-gel luminescence to look at full-length protein. Up on the right is a gel that's showing representative in-gel luminescence, and we did basically a dose response of tRNA for W1282X. In cells and looked at the amount of protein that's CFTR protein that's rescued. So this is the original sequence, and this is an optimized tRNA sequence. And these are increasing amounts of tRNA. The optimized, you can see that you get much more protein. These results are interwoven. So you have the original and then the optimized tRNA sequences with increasing amounts of DNA at 3 different mutations. The take home here is that you need about 16-fold less tRNA when they're optimized to rescue the same amount of CFTR protein. And so we're really excited about that. And this is for the tRNA that suppresses with an arginine and also one that suppresses with a leucine. And when you start mucking around with the tRNA sequence, you have to be concerned with changing the fidelity of what amino acid is being charged with. And so we determined that in all these optimized tRNAs, using mass spectrometry, that they're actually encoding the intended amino acid. And that's what these results show here. We had hex cells that we transfected in with our tRNAs, a superfolder GFP with a stop codon, and then using mass spec, we can look to see which amino acid is incorporated. So arginine goes in the optimized, leucine goes in with the optimized, and it's the glycine, which results I'm not showing here. The one where actually we converted that it did not work with is tryptophan. Our tryptophan was a really terrible tRNA, and we wanted to improve it. And we used the same kind of like workflow that I showed you for leucine and arginine. But when we got done with it, we did improve its ability to suppress, but we also made it an arginine suppressor rather than a tryptophan suppressor. So it's really important to do these mass spectrometry studies at the end when you change the sequences of tRNAs. And then the last bit, actually, I'll just pass over this. I don't have time to do this, but we're coming up with ways to basically decrease the size of the delivered tRNA by using what we call picovectors. Because the tRNA gene is so small, we can put them into really tiny DNAs. And if anybody wants to discuss this, basically we've made the smallest expression vector that we know of to date. So we found that a 200 base pair, what we call picovector, is able to express a tRNA when delivered to a cell and effectively suppress a PTC. So what I've shown you here today is that endogenous CFTR transcript abundance and channel function can be significantly rescued by tRNAs, which appears to be limited only by the delivery efficiency of the tRNA into the cells. We found that one tRNA sequence can rescue functional CFTR encoded from endogenous genes and harboring the most prevalent CF PTCs. We have successfully made tRNAs more efficient at PTC suppression by nucleotide substitution. And then what I didn't show you, because I didn't have time, is that we have developed a picovector technology for efficient expression of tRNAs. And we think that these really tiny picovectors or be easily delivered to cells by lipid nanoparticle formulations. And once in the cell, can freely traffic into the nucleus where they need to be in order to be transcribed. Future directions is determine how many CFPTCs can be functionally rescued with one tRNA. Obviously, then to figure out delivery, pairing our technologies with technologies developed by other individuals to get them into the target tissue. So the key players for this work was a very talented graduate student in my lab, Wuri Ko. She doesn't like her picture taken, and so this is her turning around after I yelled her name. She's very into drinking coffee. This is her dinner. These are all iced coffees. And then the postdoc, Joe Porter, pictured here, he does not drink coffee. And so you can see him laying deer on the ground. And these are all of the people that in my lab that have contributed to the study, and black are the main players, and then all of my collaborators, and also the funding sources. And with that, I'll take any questions.
Okay, outstanding, John. Excellent talk. We already have, I was going to ask one right away, but since there are already questions in the chat or in the Q&A, I'd rather go to those. So first question is, beautiful work, John. Quick question. What happens to natural termination codon suppression? I think that's what TC stands for.
Yep.
When you optimize the ACE tRNA and suppress NMD, is there a risk of creating new disease or problems?
So when we optimize our tRNAs, what we've done is effectively made it so that we have to deliver less ACE tRNAs to get the same result. It does not detarget natural termination codons. And so if you deliver more of our optimized, the propensity for reading through natural termination codons will go up. So really the therapeutic window is getting therapeutic threshold of PTC suppression and not NTC read through.
And on mass spec analysis, have you done mass spec analysis to sort of look to see if there's natural read-through in response?
So we did ribosomal profiling, and we shown that the very little NTC read-through has been done, and that's in our Nature Communications paper if you want to look at that. And it turns out that was actually the study that convinced people to invest in this technology to make a therapeutic. And we're actually working on different ways to identify potential NTC read-through.
Excellent. Next question. Thank you for the very interesting talk. The underlying mechanisms or sequences among the quote winners and loser ACE tRNAs, which are displayed in your 2019 paper slide, because mutation introduced into the anticodon is the same, the difference must be derived from sequences other than the anti. I actually had the same question. So
Yeah.
how is it working? How do you determine winners and losers? What's going on?
So so people are now using our data, I presume. The actual functional screen is a framework for knowing which nucleotides can be changed and which ones can be, not changed. And so we actually, in our very limited ability for bioinformatics, could have predicted which ones were winners and which ones were losers. And that was why it was so important to do the functional screen. And that's why we're always moving forward with functional screens. We now, even with now our optimized sequences, we know what areas of the tRNA that we can change and which areas we cannot change. And then we're moving forward also now doing more of a kind of bioinformatics AI approach to better get more sequence landscape.
Excellent.
Answers, functional screens are important.
I agree. Two more shorter questions. This one's the holy grail question. Do you think it's possible to engineer an ACE tRNA that only recognizes the PTC and not the actual stop codon?
No. I don't think so unless you introduce other technologies on top of it. I do not think it's possible. Just an ACE tRNA alone, no, I don't think so.
I don't disagree. All right. Technical question and a reasonable one. Do you have any idea about the amino isolation efficiency of your ACE tRNA? How many of them get?
Yeah, that's a great question. So in our screen, the ones that won and the ones that failed, we don't know why that is the case. And so it could be absolutely one of the factors could be the acylation of the tRNA, the charging of the tRNA. And likely many of them are when you mutate the anticodon, which is a recognition sequence for the amino acid tRNA synthetase, that you're reducing that charging efficiency. And so we need to go back through and pick some winners, some mediums, and some losers and see if the charging is being affected or not.
Yeah, great question. I'm gonna finish with my own question and then we'll move on. Mine's gonna be real short. You made the comment, I think, on the last slide about delivery. So yep. My question is sort of regarding this viral deliveries or vector deliveries. Do you want these plasmids to integrate, or do you want them to stay like as episomes? Do you know? I mean,
yeah. So
what is the desire here?
It's a great question. So for tissues that are hard to get to, you're going to want a very durable therapeutic. So if you're going to do whole body muscle, you're going to want your therapeutic to stick around for a very long time. Some people have thought about integrating, getting enough copy numbers to do that. Using AAV remains episomal, but it hangs around for months, right? And then it would be beneficial to do that. For the lung, because we can deliver daily, it's not necessary to do that. And so we need to think about safety. And so if you have the ability to deliver daily, you do not want to make something permanent because you can't turn it off. And so you have to balance safety, durability, and all of these. And these are things that we're considering.
Yeah, I think integration would be dicey, right? So
Yep.
you probably want to be careful. All right. Well, thank you, John. That was an excellent talk.
Thank you.
And so you can probably stop sharing, and I'm going to move to our second of two excellent talks today. I would like to introduce Dr. Amanda Larracuente. Amanda is an associate professor and the associate chair in the Department of Biology, and she's actually at the undergraduate River campus here at University of Rochester. And her talk is entitled Intragenomic Conflict in Drosophila, Meiotic Drive and Satellite DNA. Amanda, all yours.
Thank you so much. Can you hear and see my slides?
You're 100%. Go ahead.
Thank you so much. So I'm an evolutionary geneticist, and we are interested in selfish genetic elements, the conflicts that they create in genomes, and then how those conflicts fuel genome evolution. And so I'm going to start off by explaining what I mean by selfish genetic elements and conflict. So selfish elements are any sequence that can increase in frequency in the genome. So they can spread in the genome, or they can increase in frequency in a population, and not because they're doing something good for the host. In fact, oftentimes they're downright harmful, but they can spread anyway. And our genomes are full of these. So one famous example of a selfish genetic element are transposable elements. So these are mobile genetic elements. They can jump around and move in genomes. And this can be a big problem if it jumps inside of a gene and it interrupts their function, or it can cause genomic rearrangements. And so these are somewhat of genomic parasites. And so organisms have defense mechanisms in their germlines to silence these transposable elements. And this is achieved in many organisms through small RNA-mediated pathways. Another type of selfish genetic element that are really overtly selfish are called meiotic drivers. And this is one of the foci of the talk today. So one of the fundamental principles of Mendelian inheritance is the equal segregation of alleles to 50% of gametes. But there can be cheaters that subvert this normally fair process of meiosis, and they can bias their transmission to more than 50% of gametes. So these cheaters are called meiotic drivers, and they can spread in populations even if they come at a cost to the host. So we find drive in the male germline, where they often manifest as sperm killers. And we can also detect drive in the female germline because there's an asymmetry in female meiosis, where only one of the four products of meiosis becomes the oocyte, and the others are the polar bodies. And so chromosomes that can bias their segregation to the oocyte achieved a biased transmission. And so this is female drive. So meiotic drive is really widespread in nature. We have empirical examples in plants, in mammals, and insects, and in fungi. And the last type of element that I want to introduce you to are repetitive DNAs called satellites. So these are huge blocks of tandemly repeated DNA sequences that can occupy really large blocks of the genome, hundreds of kilobases, even megabases. And they accumulate around centromeres, telomeres, and on sex chromosomes. And they do perform some sort of structural roles in chromosome segregation and heterochromatin formation. And they have the capacity to become selfish, especially if they are linked to the centromere, at the centromere, near the centromere, and can cause biased transmission in the female germline. We have some examples of that. And so selfish genetic elements are really pervasive in nature and in genomes. And all of these selfish elements can cause conflict that lead to really rapid genome change. So for example, as a selfish element is cheating, host mechanisms can evolve to suppress or to resist that cheating. And then the selfish element might have to evolve around those host mechanisms. And you can see how this could keep going back and forth and trigger these evolutionary arms races. So this is the process that we're really very interested in my lab. And we study Drosophila melanogaster as our favorite model system. And we have a system that involves a meiotic driver in males and a satellite DNA. And so this is the system that I want to introduce you to. So this is called segregation distorter, or SD. And the way that this works is that males who are heterozygous for SD and a wild type chromosome transmit SD to over 95% of their progeny. But females who are heterozygous for the SD and the wild type chromosome transmit SD fairly to 50% of their progeny. So this is a very strong male-specific meiotic drive system. And we know a lot about the genetics of the system. It's autosomal. It's found on natural second chromosomes and natural populations of Drosophila melanogaster. And the genetics are quite complex, so I'll just simplify with a schematic. So this is an example of a schematic of a second chromosome that is an SD chromosome. The centromere is here, and this dot, and then this gray shaded region is the pericentric heterochromatin. So there are two main components to this drive system. The main drive locus is on the left arm of the second chromosome, and its target is on the right arm of the second chromosome in the pericentric heterochromatin. There are multiple alleles of the target that are polymorphic in natural populations. SD targets wild-type chromosomes that have sensitive alleles of the target, and it kills their sperm in spermatogenesis. Now, it turns out that SD is not just a selfish gene. It's actually a selfish co-adapted gene complex. So there are multiple other loci that you can find on the second chromosome that, when they're present with SD, they can enhance drive, they can strengthen drive. And so SD chromosomes tend to pick up chromosomal inversions that suppress recombination and then keep this sort of complex of all the genes that contribute to drive linked together. And then there are different flavors of SD, different haplotypes of SD that are polymorphic in natural populations, and they might have different enhancers. So we don't know the molecular identity of any of those modifiers of drive, any of the enhancers, but we do know the identity of the two main components of drive, the driver and the target. The driver is a partial tandem duplication of a gene called RAN-GTPase activating protein, or RAN-GAP. And this duplicate gene encodes a functional, active, enzymatically active, but truncated protein that ends up being mislocalized. And I'll come back to that point later. Now, the target is not a gene at all. The target is a large block of tandem repeats. These are satellite repeats called responder. We abbreviate it with RSP. So responder consists of a dimer of two related 120 base pair repeats, and then that dimer is then tandemly repeated in the heterochromatin. Now, what's interesting about this satellite is that the abundance of the satellite correlates with the sensitivity to drive. So if you have a really big locus, you're very sensitive to drive, but if you have a smaller locus, then you can escape drive. And if you have a complete deletion, you're an SD chromosome. So SD has no responder at the target site. SD is a sperm killer. So ordinarily in Drosophila melanogaster, wild type sperm have these nice needle-shaped sperm heads. But if you look inside the testis of an SD heterozygous male, what you might see is that about half of these sperm have these nice needle-shaped sperm heads, but the other half don't. They'll have some defect. In this case, they're these round blobs. So presumably these are the wild type, and now we can show that, spermatids that are going to be lost, and these are the SD spermatids. And so somehow SD is disabling these wild type sperm through some chromatin condensation defect that we still don't really understand. So what I want to do with the rest of the talk is tell you about some of our work on the drive phenotype. So looking more at that chromatin condensation defect. And then I want to talk about what we're thinking about when it comes to mechanism. And just to let you know now, we actually don't know at all what the mechanism is. But along the way, as we're trying to think about how this system is working, we're learning a lot about how satellite DNAs are regulated in the germline. So first, I want to introduce you to the Drosophila melanogaster testis and spermatogenesis. So melanogaster testes are these coiled tubes, and spermatogenesis occurs in cysts, and it proceeds in order through the tube. So the germline stem cells are at the apical tip here, and then there's 4 rounds of mitotic divisions, and then meiosis occurs around here. And then after meiosis, the sperm differentiate into mature sperm. And at the very end of this process, they individualize out of the cyst into their own individual sperm and are deposited in the seminal vesicle. So the visible defect in SD occurs after meiosis during this process of spermiogenesis. Normally, the sperm have to really do some incredible things after meiosis to package their DNA into the mature sperm. So the nuclei, these newly haploid round spermatids, have to shrink like 200-fold to fit inside the mature needle-shaped sperm heads. And this is at least in part accomplished by swapping out histones for these small sperm-specific histone-like proteins called protamines. So the first visible defect in SD heterozygous testes is during this histone to protamine transition. And some authors like Dan Hartle and others had proposed that there is a defect in the histone to protamine transition in SD. There's some genetic evidence for this from Jana McLean's lab, and then we showed this in a collaboration with Benjamin Lopin's lab just a couple of years ago. So I'll just highlight one of the results there. And so this is showing a wild-type sperm bundle around the time of individualization. So this is at the end of the process of spermiogenesis or sperm maturation. And this is a wild-type bundle. This is an image from Xiaolu Wei's, who is a former graduate student in my lab. And so here we have DAPI just showing the sperm heads. This is in a protamine RFP tagged fly, so the protamines are red. And then we can look at the merged image and see that at this time of spermatogenesis, the wild-type sperm should have protamines. But when we look at a similar stage in SD heterozygous testes, half of the sperm have protamines, but the other half do not consistently have protamines incorporated. And so, and we also can show that these indeed are the responder-sensitive sperm. And so there does seem to be a defect in this histone to protamine transition in SD heterozygotes. But why is this happening? What is the mechanism? The short answer is we don't know. But a lot of what we know about this, or what we're thinking about this, comes from really beautiful work in Barry Genetsky's lab at the University of Wisconsin in Madison. And the Genetsky lab focused on the role of the main driver in this whole phenotype, and that is, remember, the driver is a partial tandem duplication of RAN gap. So ordinarily, RAN gap is tethered to the cytoplasmic face of the nuclear envelope. And then together with its corresponding guanine nucleotide exchange factor, which is in the nucleus, in flies we call this RCC1, they establish a gradient of active and inactive forms of RAN across the nuclear envelope. And this is the so-called RAN cycle is important for many cellular processes, but is probably most famous for its role in RAN-mediated nuclear transport of proteins and some RNAs. So one of the important results that the Genetsky lab had was that that truncated duplicate of RAN gap, the SD RAN gap, is missing some sites that are important for its intracellular localization. And so it accumulates inside of the nucleus. And so if you have RAN gap inside of the nucleus, that throws off the RAN cycle. And they showed that there is a defect in nuclear transport. And so they proposed that there's now something probably important for responder packaging that can't get to where it needs to go. And this might be the cause of drive. So this work was really, truly groundbreaking in our field, but it also leaves open a lot of questions. So one is that the RAN cycle, this process is really important for all cells. So why is this phenotype so specific to spermatogenesis? And what does this have to do with a satellite DNA? So these are some major open questions. So we're approaching this question, but now from the perspective of the satellite DNA and thinking about mechanisms of drive. And if we want to know more about what is causing this phenotype, this chromatin condensation phenotype because of its satellite DNA, we need to know a lot more about how satellites are regulated just generally in the germline. We know so little about satellite DNA regulation. And one of the reasons why we don't know very much about it is because they're highly repetitive elements and they're extremely difficult to work with. And so we and others have been using long-read sequencing technologies like Pacific Biosciences and Oxford Nanopore to assemble satellite DNAs and other repetitive regions of the genome, build better genome assemblies, so now that we have something to work with. And so we've done this for the responder satellite. And here, so our reference genome has a sensitive allele of responder, and so it has one of the bigger loci that will get targeted by SD. So we have now the detailed organization of this this locus. It's about 100, 150 kb. A large part of it is just tandem responder repeats, that this is what this blue is representing, but there are transposable elements in here too. So now that we know the detailed structure of the satellite, we have this platform for doing functional and evolutionary genomics. So we want to come back to this question about satellite regulation, and we're asking this a little more broadly instead of only in the context of SD. Now, satellites are packaged as heterochromatin. And so a lot of what we understand generally about the germline regulation of heterochromatin is coming from recent work on small RNA pathways, where chromatin regulators are guided to specific targets by small RNAs. And in flies, this is best studied in the context of how piRNAs silence transposable elements in the female germline. So piRNAs use both transcriptional and post-transcriptional silencing mechanisms to prevent TEs from jumping around and making a mess in genomes. They can target the TE-derived mRNAs for cleavage, and they can also direct heterochromatin formation and silence TEs transcription at the chromatin level. So we reason that transposable elements and satellite DNAs have some things in common. So one, they can both cause genomic instability, and second, they are both highly repetitive sequences. And so we wondered whether satellite DNAs might also be controlled by the piRNA pathway. So indeed, our lab and others have identified small RNAs that map to satellite DNAs. And we can see these in both testes and in ovaries of flies. So here I'm showing you the size distribution from small RNA-seq data of responder-derived small RNAs. And we know that now most of these have the signature of piRNAs. And so most of these are actually piRNAs. We don't know where they are coming from at this point, just looking at the distribution of RNAs. So that's something that we wanted to know. And in Drosophila melanogaster, most piRNAs originate from piRNA clusters. So these are discrete loci of the genome that usually have fragments of transposable elements and other types of repeats, and they produce the precursor transcripts that are then processed into the piRNAs. And there are two types of these piRNA clusters. These unistrand clusters that have promoters that kind of look like genes, and then these dual-stranded clusters that don't have distinct promoters, or many of them don't, some of them do actually, and they're transcribed on both strands. So these dual-stranded piRNA clusters are really interesting because they're transcribed in a heterochromatin-dependent fashion. So we want to know where do the responder piRNAs come from. So it could be that there's a couple of repeats that are in one of these piRNA clusters, and they get included in the precursor transcript and made into piRNAs. Or it could be that the satellite itself is the source of the piRNAs. And so to ask this question, we had done some RNA-seq and quantitative PCR and northern blots using satellite probes to try to understand what these transcripts look like. And so this is, on this northern blot, we're showing 3 different genotypes that have increasing number of responder copies. And you can see that we get the smear of transcripts, and the darker smears and the genotype that has the higher copy number of responder repeats. And we can also do this a little more quantitatively by using quantitative PCR to estimate copy number in the genomic DNA for responder. And then we can quantify the transcripts using quantitative RT-PCR. We see a correlation between the abundance, the copy number in the genome, and the abundance of the transcripts. And so this suggests to us that the satellites themselves are producing these transcripts, rather than it's a couple of copies being inserted in a piRNA cluster. when we look at the RNA-seq data, this is showing small RNA-seq data. We can see they're not really highly expressed, but when we map those reads back to our assembly of the satellite repeat, we get reads that map well to both strands. So this is sort of reminiscent of a dual-stranded pi-RNA cluster. It seems like the satellite DNA is probably the source of the pi-RNAs, and they kind of resemble in many ways these dual-stranded pi-RNA clusters. So dual-stranded pi-RNA clusters are very interesting because they have this non-canonical transcription that's regulated by the rhino deadlock cutoff complex, or RDC. So we usually think of H3K9 trimethylation as a hallmark of silent chromatin, but in this case, it's actually required for initiating transcription at these dual-stranded pi-RNA clusters. Rhino is an HP1 pair log, and it directly interacts with the H3K9 trimethyl repeats, and it recruits cutoff and deadlock, which together with moonshiner license transcription from these loci. So when you look at mutants or germline knockdowns and for these four of rhino, deadlock, cutoff, and moonshiner, we can see that you lose pi-RNAs from these sort of classic pi-RNA clusters. One is called 42AB and one is called ADF. These are dual-stranded pi-RNA clusters. And the darker purple is negative log 2-fold change in the germline knockdown over control. But these unistrand clusters that are not regulated by the rhino deadlock cutoff complex, there's no change. So when we look at the satellites, in these knockdowns, we can see that we lose pi-RNAs for the responder satellite, and this is also true of other complex satellites. So it does seem to be regulated in a similar way as these dual-stranded pi-RNA clusters, at least by the same machinery. And we don't actually know what the responder-derived pi-RNAs are actually doing in the ovary, but we have evidence that they're important for establishing heterochromatin in the early embryo. We know even less about what these pi-RNAs might be doing in the testes. We could see that they're there. And recent work in Alexei Erevin's lab has shown that the RDC complex is active in testes, and so are the complex satellite pi-RNAs. So our hypothesis is that these pi-RNAs are important for packaging the satellite DNAs in testes. And if this is true, then one thing we're wondering is if SD interrupts this packaging process of the satellite DNAs from responder, at least. And maybe this is a disruption in either the biogenesis or maybe the localization of the targeting of the pi-RNAs. So a number of authors have proposed a model where these small RNA pathways are involved in drive. And there's some precedent for this from Robert Reenan's lab at Brown. So they made an aubergine knock-in on an SD background. Aubergine is an important gene in the pi-RNA pathway, and they show that it enhances the drive phenotype, suggesting that there's some relationship between the pi-RNA pathway and SD. And so we want to test this hypothesis about the role of these responder-derived RNAs in drive. And one of the first things we wanted to look at is the localization of the responder transcripts. And remember, I said the SD-RAN gap is mislocalized pretty grossly in these cells. And so we wanted to take a look at the responder transcripts. So this is a RNA fluorescence in situ hybridization image of a Drosophila testis. So here's, just to orient you, this is the fly testis down here. We're up at the tip of the testis where you have your germline stem cells, mitotically dividing cells, and early cells pre-meiosis, these spermatocytes. And I'm showing you in this magenta color is the responder probe, and in yellow is 1.688, which is a completely different kind of satellite DNA. It doesn't, to our knowledge, have anything to do with this SD system, but it's sort of a reference for us, a control, because at some level, we have to be able to explain why responder is a target and how it might be different from other satellites if it is. So what we see is that responder is, we can detect transcripts early on in premeiotic, all the way up at the germline stem cells and spermatogonia. And we also see some just before meiosis, some transcript. And 1.688 is similar, except we don't see as much expression very early. So this is one difference, and we're wondering, it seems interesting, but we don't know what that means yet. So when we look in an SD heterozygous testis that's driving, and we look for large differences, we don't really see any, at least at the level of localization. So we don't see this gross mislocalization like the Gnetsky lab was seeing with RNGAP. So there's nothing really obviously different about where those RNAs are. But what about a problem with making the pRNAs? So to ask that question, here I'm going to show you RNA-seq data. So we've done total RNA-seq and small RNA-seq and many genotypes, but I'm just going to show two here. This one is the SD heterozygote that is driving. And then we have a copy number control because remember we've observed there's a correlation between the copy number and the expression level. So we have to check that. And so if nothing is really happening here, our expectation is that SD and this copy number control will have the same abundance of transcripts. But if SD is disrupting responder, say, pRNA biogenesis, we might expect to see fewer of these responder-derived RNAs in the heterozygote. And that's what we see. So we do 3 replicates of RNA-seq in each of these genotypes, and we do see significantly fewer pRNAs coming from responder in the SD heterozygote, which is exciting. But we'd like to know if this really has to do with drive, because this genotype here, this is the deletion of the responder locus. So it's a good copy number control, but it's not a great control for the genetic background because there are some differences between this SD chromosome and this chromosome. And so to make a better control for this, we made a revertant SD chromosome. So we made a deletion of the second copy, the duplicate copy of that RNGAP gene on an SD background. So now our genetic background is the same as this SD chromosome, but it has a deletion of the RNGAP locus. And so it doesn't drive anymore, or it drives very, very, very weakly. And in this revertant, we almost completely recover the abundance of small RNAs coming from the responder locus, suggesting that this disruption of pRNA biogenesis might actually, or abundance, might have to do with drive. And so we're really excited by this result. And it's consistent with this model of these small RNAs being important for packaging the satellite. Right now, what we're doing is we're overexpressing pRNAs from responder specifically, and we're knocking down pRNAs from responder specifically to see if we can rescue and then phenocopy drive in a sensitized background. But we're not quite ready to show those data yet. Okay, so I just want to quickly summarize what I told you about today. I told you about this sperm killer that we work on called segregation distorter. It's a male meiotic driver, and it causes chromatin defects in wild-type sperm that have a big block of this particular satellite repeat called responder, and we can show that the defect is in the histone to protamine transition. And we're wondering about the mechanism, and as we're trying to understand the mechanism, we're learning a lot of really interesting basic biology about the regulation of satellite DNAs in the germline. So we've learned that responder is regulated like a dual-stranded pyranate cluster, and that in the one genotype that we're studying, this one SD chromosome, it does seem to disrupt responder small RNAs. And the last thing I want to leave you with is that there are a lot of different drive systems and different species and different loci that are independent, but many of them have a similar phenotype. They have this post-meiotic chromatin defect, or something is going wrong after meiosis. And when we look at the systems where we know at least a little bit about them, some themes are starting to emerge. One of the themes is that these systems tend to involve heterochromatin. Another is that there's a lot of gene duplication involved, or duplicated sequences rather. And the third is the involvement of small RNAs. So there's this system, a suppressor stellate and stellate, where suppressor stellate is a multi-copy locus on the Y chromosome that produces pRNAs that suppress this X-linked stellate locus. And we think that this is a relic meiotic drive system where the suppressor fixed. And then there's another system in Drosophila simulans where this X-linked driver is suppressed by autosomal loci that produce endogenous siRNAs. And so we're wondering if segregation disorder is going to fit in with this theme, and we're really excited to see what generalities there might be here. Okay, with that, I want to thank my lab. Most of the work that I showed today was work from a former graduate student in the lab, that's Xiaolu Wei, and I have a fantastic group. I also have really fantastic collaborators. I collaborated with Benjamin Lopin's lab to do the SD chromatin phenotype work. And this project is supported by an NIH R35. And if we have time for questions, I will take some.
Okay, outstanding, Amanda. Thank you. That was an outstanding talk. I asked that people type questions into the Q&A. I will take the first two because I can. I'm the host. Amanda, I have one question that I think will be straightforward for you to answer, and the other one I'm not so sure. So the question is, this responder region, is that conserved in other Drosophilids, or do they have a responder-like sort of sequence? And then my second question, which I think maybe is a little bit trickier, is if you look at Drosophila in the wild, like, not as lab strains, I don't know if this is known, but how does it behave? Is this something that lab conditions are influencing in some way? I mean, do we know what it looks like? So what are your thoughts?
Yeah, those are really great questions. So your first question about conservation of the responder satellite is this canonical version of the responder repeats seems specific to Drosophila melanogaster. This is interesting because it's a dimer, so these two repeats are related to each other. And if you look at the amount of divergence between them, you can infer that actually it's an old repeat. So it suggests that this repeat might have been lost in other species, which makes us wonder why. And your second question was about, is this something weird about lab conditions? I mean, geneticists have been studying this system since the 1950s, and it is this like weird dominant gain of function thing, but it actually is in all wild populations, almost all wild populations of Drosophila melanogaster across the globe, or its distribution, you can find SD. There's a ton of variation in the phenotype and the strength of drive, and I'm not sure, necessarily how much of that has to do with what's going on in the lab. Certainly some of it could be, but it's because there are modifiers of drive all over the place. We have to control genetic background. Very, very carefully, because when you are talking about a pathway that involves RAN gap, and you think about loci that might have the ability to affect, variation that might affect the function of RAN gap, that it's huge, right? Because there's so many things that interact with the RAN cycle. So, yeah.
I can envision a lot of things modifying that transcriptional regulation at the loci itself. There are tons of proteins that would influence that. I have another question. So I have never heard of rhino cutoff and deadlock. This H3K9 complex that you said turns on transcription. How does it do that only in that locus and not another H3K9?
Oh yeah, it's so interesting. So a lot of what we know about this came from Peter Anderson's work, and there's some really interesting work from Julius Brennecke. It's hard to, if you look at these pRNA clusters, these loci, there's not a whole lot that is obvious that distinguishes them from sequences next door that don't produce pRNAs. They're all enriched in these H3K9 trimethylation. So we don't really understand at all for the satellites right now what would make this one, a pRNA cluster, and that one not a pRNA cluster. And pretty much all the complex satellites that we've looked at are behaving kind of similarly. But for other loci in the genome, recently the Brennecke lab had discovered a zinc finger protein that is involved in determining which clusters are going to be active pRNA clusters. So I think there's still a lot of questions about how that is determined. It's really fascinating, I think.
Now you got me interested talking about zinc finger genes. We're interested too. So I do have a question here. Do the smRNAs targeting The responders show U bias at the 5' end. What about a ping pong signature?
Yeah, yes. I just tucked that in the corner of the slide. I didn't emphasize it. Yeah, these do the majority show the signatures of pRNAs. They have the 10 nucleotide overlap and the U bias. And more so in the ovary than in the testes, which is typical of pRNAs and Drosophila, melanogaster at least. Good question.
Okay. All right. Well, it looks like there's no more open questions. So I want to end by thanking our two speakers. Give us a virtual thank you to all of them. And I want to make one last announcement. The next RNA Collaborative Seminar is on February 14th, hosted by the RNA Maxi Group at the University of Wisconsin-Madison. So let me make sure I'm not missing anything else from the chat. I think we are good. Thank you all, and we'll see you, I guess, in about a couple weeks.
Thanks, everyone.